Utida. She is a postdoc at the Institute of Geosciences of the University of Sao Paulo in the area of isotopic geochemistry and paleoclimatology. She holds a PhD in sciences at the same institution in the area of geochemistry of exogenous processes. Also Master of Science from the University of Sao Paulo in the course of geochemistry and geotectonics, with emphasis of paleo-environmental reconstruction. She has experience in the area of isotopic geochemistry, paleoclimatology, and geochronology in speleothems and lake sediments. She works mainly on paleo-environmental reconstruction of tropical regions and isotopic geochemistry. And today, Giselle will give us a talk on multi-proxy studies. Please, floor.
Thank you, Nella. Hello, everyone. Thanks for coming. You were great. You're here by the end. And today, I'm not going to introduce you something new because it's working.
Yeah.
Because we already see many things here, like introduction about speleothem research in Brazil and South America. We already talked about speleothem formation, radiatorium dating, like the basics on speleothem science. We also talk about oxygen isotopes and carbon isotopes, value temperature, that's compared to the Earth is new proxy, trace elements with Kettling and petrography, and finally today magnetism, that is also something new in the camp of satellites. And finally Matthias that introduced us how to use models related to our data. And now I'm going to join everything and send you a message about how to use everything together, all this data together, how we can do it, and why you have to do it. Multiproxy. So, we already do multiproxy studies. All of you do dating, oxygen isotopes. Usually we do trace elements, we count layers, so we do many things using one sample. But have you think about if this sample is enough to reconstruct your environment and your climate? Think about this. I want to give you a message, not exactly a technique, okay? So what are the advantages of using multiproxy? So we can complement our interpretations or confirm our interpretations. As we already see with, for example, Roger that talked about the vegetation, and we use also strong suit to confirm that it's really vegetation while we are watching in our carbon. Then we want robust results because we want to have good results. We don't want doubts. We want answers. And we also can reach new parameters like the temperature. But if you use multi-archives, think about your cave. We have stalagmites, but we also have many other boxes in your cave. How to use them? Anyone here works with someone else in the cave besides stalagmites? Everyone uses stalagmites? What do you want? What do you use it?
Looking for cave sediments and aerosols, but to diagnose things.
And also stalagmites. Which one? Someone else? Ice and stalagmites. So, just two people?
It's not in the cave, but I work with corals.
Corals and stalagmites. Good. And
alluvians and stalagmites.
and stalagmites. One more person.
Lakes and stuff.
Lakes and stalagmites.
It was the same, like lakes.
Oh, good. So we are using mold archives and mold proxies, not only one sediment or one stalagmite, but many things to tell one story to make it more complete. Whoops. So let's keep the other one. So we have many things inside a cave, and how to use those things. We have stalagmites, but we also have classic deposits, as you said. We have red guano, we have fossils, and we have also archaeological evidences. But sometimes we have other things that maybe we didn't pay attention before in our cave. So we have to think about the whole environment, not just looking for stalagmites. For example, we have many kinds of different sediments, depends on your cave and the size of your cave. You see, big plastic sediments or even smaller ones, doesn't matter. We can think about our questions. And we also have stalagmites. So how to connect those archives in our cave. We also have some fossil remains. And what is the relationship between the significance of our fossils and our stalagmites? They were there at the same time. How was the climate? This animal was there before, after, why it died. So we can construct a better picture of the whole cave and environment outside. We also have other kinds of animals inside, and also archaeological remains. How to connect them? I'm asking more questions than answer things here, and that's what I want to do. I want you to think about your data, not my data. Okay, and we also have sometimes one cave with stalagmites and another cave that is close but in the same system, in the same bedrock, in the same climate system, that have other things that we can connect them. This is you remember the picture with me inside the guano. That's the pile of the guano, see? It's so big. And we can connect our archives, our stalagmites and the guano and the sediments and many other things that we can find inside the cave. So, nothing is new here. You already see this paper here before twice. And I want to show you again, but not exactly to interpret the data, but to think about how I did this work. What I used here, which archives and which process. So, this area is in Northwest Brazil, and I use stalagmites to reconstruct climate and other archives. In the beginning, this is the second time today, as Matthias said. Well, you're going to show you again. So, in the beginning, she could reconstruct climate using only oxygen isotope and stalagmites. And okay, it was a good work, and how we can do more? Then, in my paper 20, I present the carbon isotopes. But the carbon isotopes is not similar to oxygen. What's the meaning of this? How to interpret the carbon only using oxygen? I don't know. I had to find more things to explain this. One thing that I checked was the bedrock signal. And for the last 4,000 years, the carbon was very similar to the bedrock. Okay, I need more things to explain this. Because the mother conditions is no soil on the surface of the caves. So how the signal was negative? If you will already know that the negative signals for carbon can be from the soil. How to explain this? So I test another proxy for the stalagmites. I test the strontium. And you already know the story, or Audrey just talk about more about carbon. So I'm going a little bit in this. So the strontium comes from different sources. One can be the bedrock. Then I test the bedrock, and the bedrock has here it's low values, and here it's higher values. So the bedrock has lower values compared to the stalagmite signal. But the stalagmite signal, when we look to strontium, was very similar to oxygen. Okay, maybe there is a meaning, but I'm trying to find an explanation for carbon. Then I try to find other things inside or outside the cave. Outside the cave, there's no more soils, but we have some bases of soils. Sometimes you can find soils in the fissures, and some areas we have a little bit of soil. So I check the signal of strontium of these soils. And I also check the signal of sediments inside the cave. This is the same cave. The condition above this cave is the same, but it's a different cave where we find the stalagmites. But it's very close. It's just 1 kilometer apart each other. So it's the same system. And the signal, the strontium signal of this sediment is the same of the old soils above the cave. So I can say that during this period with high rainfall, the soil was eroded and transported inside this cave. Okay? And it means that if the strontium has the same signal inside and above the cave, which was the influence in our speleothem, how can I use this to answer the questions I have in my carbon? So, as I have the strontium from bedrock and the sediments and soil, we can calculate how much is the bedrock contribution in your stalagmites. It's just a neighbor calculation, very simple. And here you have the bedrock contribution. When the signal is close to the bedrock, you have more bedrock contribution in your stalagmite. When the signal is more positive, it's closest to the soil. It means that during this period, we have more rainfall and more soil contribution here. That's why it's negative. After this event, this abrupt event of reducing rainfall, we have a big change in carbon that is also present in strontium. And this moment, that soil contribution was really not present here. What's the meaning of this? Anyone have an idea? Do you want to guess?
Again, please.
Do you have an idea why there's no more a positive signal in the last 4,000 years here?
Because the soil is over.
The soil was, everything it was eroded, and then start this condition. This strong rainfall just eroded the soil above the cave and transported it inside the cave, and then the stalagmite signal changed. Now we have this carbon isotopes closest to the bedrock signal, and also strontium stalagmite closest to the bedrock signal. That's why we can interpret carbon, because we use more than one proxy. Only using carbon isotopes, I cannot tell this. But I still have questions. Why I have guano above the sediments? It was wet and becomes very dry. And now I have guano. The bats appear in the cave because it's dry. I don't think so. Maybe they were already there. So we also dated the sediments and also the guano. The sediments had really good ages compared to this version of the stalagmite. The sediment was only there when it was wet. And then the guano just appeared with dry conditions. Yes, you already know this. I checked the ages of guano also. And the ages of guano just appeared. Guano was just preserved when it's dry. Because when it's wet, the water inside the cave just removed and eroded also the guano. It could only be preserved when it's dry. And this gives us another information. It confirmed that it's dry. Otherwise, if it was wet, we would have erosion inside the cave and transport of sediments and also guano. So we won't have this there. The guano can also give us some answers, but it's still more, we have more questions than answers about this because we don't know what's the meaning of the mineralogies, how it's going to be formed, how is the influence of climate here, but we are still keeping studying this guano and finding more answers when we relate this with climate. We reach more answers here. Okay, same. So, after that, comparing with other archives outside the cave, this is two regions, the data of two regions, very far, but it's still in the same biome. It's still Caatinga, it's semi-arid region. And these two data are palynology data. And they are telling us that the vegetation changed from wet conditions to dry conditions in both. Here we have forests and less forests. And here is the savanna poly. And exactly at the same time, when we get dry conditions, the vegetation also changed. So during the wet climate, we have more vegetation and soil above the cave. Then, during the climate change, when it changes to dry conditions abruptly, it becomes dry and the vegetation changed. And that was the moment where the Cerrado here in Brazil, it was retracted because it was expanded here during this period where we have more wet conditions. And here we became drier and the vegetation changed. The biome here changes. Okay? So we just have this because we use multi-proxy and multi-archive. We cannot answer all these questions only using stalagmites. We have to use other things that inside and above and around the cave to answer all our questions about paleoenvironment and paleoclimate. So we have to open these boxes. We cannot stay just in the stalagmites if you want to reconstruct climate and environment. So when we think about the human societies and the impacts of climate and also environmental change, how can you use a multi-archive approach to talk about it? I'm still working in Northeast Brazil in the same area, but talking about the last 500 years, the same area, we have oxygen isotopes and stalagmites. So, okay, it's one proxy. And what's the meaning of this? We have some drought events, okay, we don't have other proxies to answer human impacts in this area. The archaeological remains are not really good, it's not present. Our meteorological monitoring, it's not very long. How to do it? How to compare and talk about the impacts in this region. Then we have to go beyond the cave, much far from here. In this case, I use historical archives. It's not related with the cave, it's related with another subject. We are going to history, we are going to other areas of studies. But fortunately, we have people that are studying this, and they collect data from many papers and many historical archives. In this case, those were the people that arrived in Brazil around 500 years ago, and they describe the environmental and the climate here. And they describe the drought events in this region. So this is the first record of drought in Brazil. It was in Northwest Brazil because it was the first area where they arrived. And fortunately, it's around the same age. So we haven't recorded in this stalagmite. And we also can identify other droughts. However, sometimes your age model is not really good because you don't have good ages. Your error can be large. But look at this interval. We don't have stronger drought here. And when we go to the historical records, we can see that this interval also doesn't have large drought events and also very few drought events. It means that, wow, maybe this report is right. Yeah. Then we can keep connecting our stalagmite droughts and the historical events. So, yeah, I think it's going to work. But we have to be lucky to find a good stalagmite to do this. Well, changing the continents and the hemisphere. Another example in China. They also studied the last 500 years. And studying the Dayu cave, tell me if it's right. Dayu cave? Okay. They find a good Speleothem with good ages, covering the last 600 years. And how to connect it historical events. Here, they were more lucky than me because the historical records was written inside a cave. So sometimes you just have to look around you to find the tips, you know? And this records was a bit dated. So you have the age and the dates according to their calendar in that period. So I have no words to say, could not be better. And they check it in the stalagmite. So they do this oxygen isotopes, the trace elements, and the carbon, and they check that during this periods, and gray here, that is the droughts recorded inside the cave. You have higher oxygen isotopes. Not all of the higher oxygen events here was recorded in the cave, but maybe it was not strong enough because we don't really can correlate this with precipitation and the impacts. The structural calcium results was also correlated with these results, and the carbon has a strong impact also. So the vegetation also suffer during these drought events. You can see this, maybe it's stronger than the Strauss-Uriel reports. And then they, of course, they try to prepare the data statistically to check these records and correlate with them, and also predict, according to the data, how could be in the future this droughts in the region. So let's change the subject again. As I told you, I'm not answering questions. Giving you ideas maybe. I hope my message is reaching you. And how can we connect stalagmites and biodiversity? Maybe using archaeological remains or fossil in the caves. But let's go to this paper. It's a cave in Switzerland, and in the beginning they start to study this cave to check the glacier history. Okay. They dated their stalagmites and they checked that between 7,000 and 9,000 years ago, it's registering the retreat of these glaciers. And as you can check here, the petrography saying that it was a calcite mediated by bacteria. Because if you don't have a glacier, well, maybe it's a little bit warmer, and then the bacteria can grow in this stalagmites. Later, in the same stalagmites, they study some specific points because, well, maybe sometimes you cannot do like every millimeter as you like to do with isotopes. And they checked the palynology of these stalagmites. They were able to find pollen grains and reconstruct the vegetation. Of course, they find it in the bacteria-mediated layers because we have more carbon content, more organic content in this part. And they could describe the vegetation using the pollen grains attached in these stalagmites. So, this is a paper from this year and the same stalagmites, and they try to check the DNA. So, it's a little bit new, but it's not the best yet. Yes. Because, well, we have 5 papers using DNA and stalagmites. And the answer, the question is, can we check the DNA here and connect with the pollen we find before? We are improving techniques. First, the first studies with DNA, they use the same micromeal we use to drill, for example, ages. So we are adding contamination to your sample, especially organic. We can find the human DNA in our renitorian samples if we check. Because, okay, we are using maybe the hands without gloves. And by using this machine, improving the technology and the methods, we can find better results. This machine, it's freeze drying mill. So you can have a piece of your sample, but here inside a tube, a clean tube, and it will freeze and magnetically milling your sample. So without putting your hands, you're going to avoid contamination. And then your DNA results should be better. So they could find in the red dots the signal of DNA of the same pollen grains they find in the palynology study. So it was not so good because the pollen has a larger biodiversity. But improving the DNA techniques, maybe in the next years, we'll be able to find more species and other kind of samples, like animals or bacteria that are mediating the precipitation in our stalagmites. And then we can connect multi-proxy and multi-archive results and find a better story about paleoenvironmental and paleoclimate changes, or how it was, how could be. So, I was very brief. Let's go to the final message for you. We already talked about advantages and multi-proxies. So we are finding a confirmation or a complement of our interpretation when we are using our stalagmites by using multi-proxies. And, of course, robust results. We are sure that we are talking about precipitation. I want to be sure I'm talking about temperature. And we also can find new parameters with new methods. But if we add multi-archives in our study, we can have different perspectives of the same environment. How it was, as Matthias said, how was it outside? And how was inside the cave? How was the vegetation? How was the biodiversity in our cave and outside the cave? Because just talking about, oh, it's raining more, okay, but sometimes it doesn't answer our questions. So we have more accurate results about our reconstruction in the past. Because we don't have a picture of the past, we have to find answers. And we can evaluate the climate impacts when it's changing, when we are finding the climate change, how was the impact during the past? If it happened, how could it be in the future? We have to study the impact to understand what can happen in the future. And maybe for people that are not from science, that's the main point, that we can explain them the importance of our work. Because if you tell a normal person outside, okay, what are you studying? Oh, I'm studying stalagmites. Hey, what is that? What's the importance of your work for normal people? We have to find answers to convince them, to tell them how we are useful for society, not just for, well, science. What's the impact of normal life? About what are we doing? So, if you understand the impacts, we can explain and convincing people that what we do, it's important. Okay? So I hope I reach my objectives, that it's giving you thoughts about what we are doing. How can you improve your work looking around your stalagmites, not only to the floor? Okay? So I want to thank you all and also the committee. Thank you, everyone. If you want questions, please.
Thanks so much, Giselle, for your exciting talk. And now time for a couple of questions. Yes, please.
Well, thank you for the insightful talk. Well, I want to ask about using the multi-property and multi-natural archival approach. So while using the multi-natural archival multi-property approach, so in different regions. So I want to understand how can we help in identify the large-scale climate text connection, especially on the large-scale study connection, and how can it assist the synchronicity, especially in the climate change, especially I'm talking about large-scale climate change.
Yeah, usually we compare different archives, for example, ice cores and oxygen isotopes, and we try to find an answer about the impacts in different regions according to atmosphere circulation or ocean circulation. And for a while, I think this is the way we do it with proxies. But using models, like Matthias showed us, maybe it's the right way to see actually our interpretations, because the models can give you a picture. You can see a map with temperatures or precipitation, and by using many different methods, I think we can reach better the global connections.
Like actually, I was talking from
You-
actually all natural archives they have different some limitations. So that's why I was asking, like on a short scale, time scale, like it mentioned. That's why I was asking on a large scale. So it might be because it could. So if we have goals like that, we have any reference so that I can follow, because I have enough certificate.
Yeah, for sure, each archive has their own limitations, especially for ages. With comparing carbon dates and uranium dates, usually the carbon is for sedimentary and the resolution is very low compared to uranium age and stalagmites. So we cannot improve the sedimentation rates, but we can interpret it and looking for your archive. For example, when we look for interpretations related to lakes, you have low resolution compared to stalagmites, and you have to understand your lake. In Northeast Brazil, I also work with lakes, and this lake was close to coast, and we have some influence of dunes, and the dunes just changed the interpretation of lake level, because during the dry periods, the dunes can block the river, and then your lake is higher level. So it's completely opposite when we think about rainfall. So we have to study each archive and look for specific interpretations that you can find there.
Any other questions?
I really liked how you connected historical records with paleoclimate records. I have kind of a very basic question. Has it ever occurred in your studies where the historical records and the paleoclimate records are pointing towards opposing information?
Well, I didn't find this, but at the same way as the lake and dunes, sometimes the results can be opposite. Can you imagine a local community that is using a river, and maybe this river is blocked by other reasons that are not climate above this region? So that for that area, it's not climate change that is impacting. So we have to take care with historical events, and take a look more in details about what we are using to compare.
Yeah, I mean, how could one do that? Like, know for sure that the historical record is actually like a much smaller picture of the greater climatic thing that's happening. How would you go about it?
Well, it's a hard question. Yeah. If you have a good description of this historical drought, maybe you can find an answer if it's a local or a global. In Northeast Brazil, for example, in the beginning, the descriptions are related more to few places. When we have more descriptions and different descriptions in a region, then maybe you can have a more regional than local effect. So we have to read more and find details about the history or about the paper you were reading, the study. Yeah, it's not really an answer, but we are trying.
Any other questions?
if we're using the multi-proxy but the results are opposite to each other, how can we do interpretation? I said if we are using the multi-proxy but the results are opposite to each other, they are not in good agreement, how we can do interpretation?
So find another proxy.
No, if they have not good agreement, for example, we use 64 proxies, but each are opposite to each other, they are not really good.
Yeah, maybe you don't have the answer yet. They can be a positive, but you have to find a reason to interpret this. Yeah, I think the best is looking for another proxy and try to relate it, or try to find another archive in the region that can answer your question. Yeah, it's not really easy, and as I said to you before, I don't have an answer. I'm just giving you ideas.
Other question? Let's thank Giselle again.
Thank you.